why I don't give you guys multiple choice questions, because I'm crappy at writing them. So,
Is this another trick question?
on accident, I'm sorry, but it should be kind of clear. So, which of the following describes anaerobic respiration? So we know it's not A because it's anaerobic. So oxygen is not used as the final electron acceptor. That is aerobic respiration, right? That's literally when we do aerobics and we're sucking eggs, we are performing aerobic respiration. So not A. It's not fermentation because, and yes, fermentation is anoxygenic conditions. So those are organisms who would normally be able to carry out aerobic respiration and get the oxygen cut off. So anaerobic respiration would be using some other oxygen-containing molecule that is not O2 gas as the electron acceptor. So we're going to talk about that here in just a second. The reason it's not fermentation is because organisms who carry out fermentation are aerobic organisms. They're facultative aerobic organisms, so they can use oxygen, but they can also go without it. Okay, so
Let's try not to put that on the test.
I know. I'm telling you, this is why you guys only have 15 multiple choice questions, and most of the time I don't write them. The ones I write, you end up getting credit back on. So D would be the correct answer. Maybe I'll start looking in the book for multiple choice questions.
I'm really bad right here.
Okay, retry.
At least someone did.
But weren't you so engaged just now in that moment? That's the point. Okay, anaerobic respiration. So it functions similarly to aerobic respiration. We still have glycolysis happening. We still have the TCA cycle. All of that is going on. The only difference is that at the very end, in the electron transport chain, oxygen is not who takes the final electron to make water. So instead, we get different products. Some of the potential electron acceptors that are not O2 gas, in the example that I give you, nitrate, sulfate, carbonate. So these things do contain oxygen, so don't let that confuse you, but they don't contain O2 gas. So even organisms who can't live in the presence of O2 gas need oxygen, remember? Because what are the nutritional requirements of every single living organism? Schnapps, right? And there's an O in that. So they need oxygen, but they have to get it in a different way. They can't be exposed to O2 gas, so they use something else. Okay. So depending on what the non-oxygen electron acceptor is, we get different kinds of products. So these are just some examples that you might see. So for example, CO3, when it is gaining those final electrons, it becomes methane gas. So our methane producing bacteria or methanogens are taking part in that route. Sometimes we see sulfate being reduced to make hydrogen sulfide or sulfuric acid, depending on what kind of organism we're looking at. We can have NO3, like I said, just kind of depends on the organism. And the yield, very similarly to aerobic respiration, is going to be variable, but it's better than fermentation. So it's better than not using anything with oxygen and not being able to carry out the electron transport chain at all. So it's going to be better than that. Okay, so that's all that we're really going to say about anaerobic respiration, other than why you should care. So a lot of organisms that fix nitrate in the soil are really participating in the circle of life, basically. So they're basically changing nitrogen either in the air or in the soil and putting it into forms that are not harmful to us, as well as making the soil more rich so that plants are more likely to be able to grow. So a lot of these are kind of a symbiosis with us. We are very much benefited by bacteria that carry out anaerobic respiration. Okay.
You want us to know those three guys?
No, I was just pointing out some examples that you might be interested in. Okay, so now we come back to pyruvate, which is the product of glycolysis. So we talked about one fate of pyruvate being aerobic respiration, anaerobic respiration. We're not going to talk about anabolic pathways. And then finally, my favorite, fermentation. So fermentation is a pathway that an organism may be able to take if it normally likes to live in the presence of oxygen and is able to use the electron transport chain, but then we cut them off from oxygen. So they can no longer use the electron transport chain because they're not able to use anaerobic compounds in order to continue that on. So what I'm talking about might be an organism like Saccharomyces cerevisiae, or brewer's yeast. So these guys are capable of fermentation. Not every organism is capable of fermentation. That's important to know. There are only some that are capable of this. And what is the purpose of fermentation? Yes, we love alcohol. Do yeast care about it? No. They're not making booze for the fun of it like we do. Instead, what's happening is they need ATP regardless of what the conditions they live in are. They don't just stop needing energy. If an organism was capable of forming an endospore, that might be one thing if they didn't have nutrients. These guys are not. So if they want to still continue their life cycle, then they've got to be able to make energy in some way or another. So they need to make ATP. Fermentation allows them to continue the process of making ATP just at much lower levels. So when we look at the pathway of fermentation, the metabolic pathway and what's happening, you'll see that one of the products of the fermentation pathway is NAD+. What is NAD+? Do you remember? It's an electron carrier. Excellent. And NAD+ is carrying electrons, it's picking up electrons in glycolysis, as well as the Krebs cycle, right? And it would normally deliver them to the electron transport chain. But when it picks up those electrons, and it can't go to the electron transport chain because there's no oxygen present to pick up those electrons, then the NAD pluses becoming NADHs, what do we do with all of that excess NADH? Where do we take it? We have to take it through the fermentation pathway. So the fermentation pathway picks up the electrons from that electron carrier. So it takes NAD, if we have NADHs that are traveling in from glycolysis and from the TCA cycle, they need to be able to drop off their electrons somewhere so that they can become NAD+. And then they can go back to glycolysis. So the reason that they can then go back to glycolysis is because, if you remember, glycolysis makes a couple of ATPs, right? So this is going to be their main source of those ATPs, is going through glycolysis over and over and over again. So they're really not going to be able to make as much ATP, right? So I like to say when we're making wine, we give them oxygen and lots of sugar for a while, and they're really happy going through their happy little pathway, and then we put the stopper in the bottle and cut off their oxygen, and the yeast turn around and give us the food. Because now we've cut off their ability to make all of that energy. So this is, I think there's a question in your homework, why did the yeast give us the finger? Because we've cut off their ability to make a crap ton of ATP. Now they can only make very, very, very little. So they can still make some, but they can only make very little. And that becomes an issue because we need energy to carry out metabolic processes. And if they're running at low energy, they're not going to be as efficient at carrying out those metabolic processes. So things like cellular division really start to slow down because they require a lot of energy. So the yeast cells will divide much more slowly, or not at all, because they just don't have enough energy to push forward. Does that make sense? Okay, great. So let's just look at that metabolic process of alcoholic fermentation. So of course there are other forms of fermentation. My mind just always goes to alcohol. But there are other forms of fermentation that we'll look at here in just a second. So the whole process of alcoholic fermentation is basically using those pyruvates that are formed from glycolysis and converting them into eventually ethanol. So there's a couple of steps that goes through in order to produce ethanol, which just happens to be the byproduct. And ethanol, like I said, the yeast could kind of care less whether or not they make ethanol. That doesn't matter to them. What they're really doing is regenerating that NAD+, because in the process of taking a byproduct, acetaldehyde, and converting that into ethanol, they're able to hand off that NAD+. So they can take the electrons from NADH and let the NAD+ go back to glycolysis so that we can keep trying to make those very little amounts of ATP. So you'll notice another byproduct of that is CO2, which is where we get bubbles from, right? So CO2 and ethanol are the two major products from that. Of course, you do not need to know the chemical symbols or formulas for any of this stuff. I just wanted to kind of show you what's going on in that whole process. So we lose basically one of these CO2s, and so we're left with this double bond to oxygen. If you like chemistry, it makes sense. Any questions about this? So, if I ask you, for example, why do yeast continue, or why do yeast carry out the process of fermentation? What is their goal in that process? Yes, the goal is to make energy, but I want to know more. So, the goal is always to make energy. But through fermentation, they have to make it through a different way. They have to use their NAD pluses to go back to glycolysis. So really the main goal of fermentation is to regenerate NAD plus so that they can make energy through glycolysis. Any questions? I wish I could have brought wine for the donuts, but some of us are underage. Oh, I actually don't have another question, sorry. Lactic acid fermentation is just another kind of fermentation. Lactate, lactic acid, pyruvate, pyruvic acid, these are interchangeable terms. And so basically this is just a shorter pathway that happens, and the pyruvate gets converted to lactate instead of ethanol. It's the same process, it's for the same reasons, it's just a different kind of fermentation, a different product. So depending on the organism and the environment that we're giving them, we're going to get different kinds of products. So where do you think we see lactic acid being formed?
In our muscles.
In our muscles, yeah. So we can actually, our cells can carry out these processes where we don't have oxygen. Let's say that you're running a marathon and you start to get short on oxygen. You still have to have a way to make energy without that oxygen, right? So your cells start to carry out lactic acid fermentation. And so we get a buildup of lactic acid in our muscles, and that is what actually results in a lot of our muscle soreness, is that lactic acid buildup. Another example that directly applies to micro is lactobacillus species, which we actually find in yogurt. So it's thanks to lactic acid fermentation that we have yogurt.
Is this 1990 plus also?
Yes. Same goal. Okay, so those are the two that I want you guys to be aware of, is lactic acid and alcoholic fermentation. But there are tons of other products that could potentially come from pyruvate and the fermentation of that, depending on the organism. Because we have such a vast variety of different organisms, it would be impossible to cover all of the different kinds of fermentation that they carry out. So I just expect you guys to know those two. Any questions? You might notice acetic acid, for example. What's acetic acid? Vinegar, yeah. So when we talked about environmental conditions and nutritional needs of bacteria, we talked about Acetobacter making red wine vinegar. When we put them into something like red wine, they'll eventually actually make acidic conditions for themselves because they like living in acidic conditions. So they'll carry out that type of fermentation and eventually make acetic acid. Yes?
Just a little kind of cool aside.
Yeah.
I had some, speaking of bacteria that grow in acidic conditions, I had some lemon juice that my kids left out of the fridge, and it started to ferment. I've never seen,
Yeah.
Carbon dioxide bubbles and everything. Never seen something that acidic.
Did you try to drink it?
It smelled good. I probably will. But yeah.
Awesome. That's really cool. Yes, yeah, that will happen. When things start to kind of smell alcoholic after a while, that is the bacteria helping along that fermentation process. Yeah, that's awesome, thanks. Okay, so this is just another table from your textbook that I think really helps you compare and contrast these items. So looking at all of the different pathways and being able to tell the difference between what the real goals are, who's involved, maybe what the final electron acceptor is, what the products are, etc. So just so you can compare and contrast those to one another as a potential study. So like I said, unfortunately, we don't have time to cover all of the different metabolic pathways. I'm sure you would love to cover every single one, but we don't have time. So what I will say is that a lot of these pathways are what we call amphibolic or bidirectional. So we've talked about a lot of catabolic pathways and not a whole lot about anabolic pathways. So anabolic pathways being those pathways that allow us to build things like proteins, lipids, etc., carbohydrates. We can actually, through gluconeogenesis, make carbohydrates. So we won't learn all of those, but just important to note that we can sometimes move backwards. So we call those amphibolic pathways. Okay. I think that is it for cellular metabolism. Yeah, this is due today. Okay. So let's get started with Lab 5. If you guys want to scrub your benches, take a little break, have another donut if you want to, and we'll get started with Lab 5.
I can't believe I'm like the only one who ate a donut. That's so people.
Everyone had donuts.
Oh, really?
Yeah. Rude.
Matthew, you're
Doing
How's
donuts.
it going?
I still want
Yeah, short-lived.
cup of soup. Cup of noodles like every time. Isn't that gross?
Have it for lunch. No, dude. Sometimes it just hits the spot.
Oh, I should probably stop this.